I'm Erin DiCaprio, virologist and food safety specialist in the Department of Food Science and Technology at UC Davis. Here we'll discuss what is a virus. Viruses are not considered to be living organisms. They are obligate intracellular parasites, meaning viruses require a specific type of host cell and a specific type of organism to be able to multiply or increase in number. Viruses also have no type of energy generating mechanism, i.e. cellular respiration. Viruses are essentially rationally designed combinations of nucleic acids, proteins, and sometimes lipids that have the ability to modulate a host cell's own machinery in a way to make additional viral copies. The central dogma of molecular biology describes the process used by all cells to maintain their genetic information and to convert that genetic information into proteins. DNA is the genetic code and has all the information necessary to make all the proteins a cell will need. The genetic information in DNA is converted to messenger RNA through the process of transcription. Messenger RNA serves as a template to build protein through the process of translation. The two-step process of transcription and translation is critical to the survival of cells. However, not all viruses adhere to this sequential process. Moreover, the virus itself does not encode all the machinery necessary to complete the process and hijacks the host cell's own machinery to generate viral proteins. There are many different types of viruses. Some viruses do have a double-stranded DNA genome. However, there are some types of viruses that have a single-stranded DNA genome. For most DNA viruses, the viral DNA genome is transcribed into messenger RNA, and then viral proteins are made by translation. Other types of viruses have RNA as their genome, and this RNA may be either single or double-stranded. In some cases, the viral RNA genome functions directly as messenger RNA and is translated into viral protein after the virus enters the cell. In other cases, messenger RNA is made from the antisense RNA genome, and then viral proteins are translated. In both of these instances, there is no DNA involved in the viral replication process. Some other RNA viruses make a DNA copy from their RNA genome, a process called reverse transcription. This DNA copy is then integrated into the host cell's own DNA, and the normal process of cellular transcription and translation makes viral proteins. Viruses that utilize reverse transcription are called retroviruses. There is much more nuance than I present here, but I give this general information to further emphasize how viruses differ from living cells. Viruses are generally nanometers in size. This makes it impossible to visualize viruses using a light microscope. In order to look at a viral particle, an electron microscope is required. Viruses are much smaller than living cells. On average, viruses are 100 to 1,000 times smaller than bacterial cells. This illustration shows a virus that infects bacteria called a bacteriophage on the surface of a bacterial cell. Viruses that infect human or animal cells are generally 10,000 times smaller than the host cell. There are two basic structures of viruses, non-enveloped and enveloped viruses. Non-enveloped viruses are the most simplistic of the two types of virus structures. Non-enveloped viruses are comprised of nucleic acid, which is coated or encapsulated in a viral protein. The viral protein that encases the nucleic acid is called the capsid. The viral capsid is formed from a protein encoded in the virus's genome, and the capsid may be comprised of multiple copies of one or several different proteins depending on the type of virus. The genetic material of viruses may be either DNA or RNA depending on the viral type. Envelope viruses are more complex in structure compared to non-envelope viruses. On the exterior of the viral particle is a lipid bilayer that is derived from the host cell's own membranes. Within this lipid envelope is viral nucleic acid, and this nucleic acid is encased in viral protein. Depending on the type of virus, there can be many other viral proteins associated with the nucleic acid or in the space between the nucleic acid and the envelope, called the tegument. Another very important structure are viral proteins that are associated with or embedded in the viral envelope. These proteins include those important for viral cellular receptor attachment, viral membrane fusion, viral envelope stability, and subsequent budding or release of new viral particles from a host cell. The morphology of the virus particle is a defining characteristic. The unique shape of each virus is contingent on the organization of the capsid or nucleocapsid and whether the virus has an envelope. Viruses in the Rhabdoviridae family, which includes rabies virus, have a classic bullet-shaped morphology when observed under an electron microscope. Ebola virus, which is a filovirus, has a classic filamentous morphology. Many other viruses have general round structures, but they can be differentiated under an electron microscope by different surface structures. For an example, the surface of coronaviruses are covered with large prominent protein spikes, which were thought to resemble a halo or crown. Corona in Latin means crown, so hence the name coronavirus. Viruses are known to infect all forms of life. There are viruses that infect unicellular organisms like bacteria, and other viruses that infect complex vertebrates such as humans. There are several viruses that cause serious disease in humans, animals, and plants. Other viral infections may go relatively unnoticed. It is important to understand that viruses are host-specific, meaning that a virus that infects a plant, for example, will not infect a bacteria or a human. In some rare instances, there can be viruses passed from animals to humans, called zoonotic transmission. This requires a change in the virus that makes it adapted to humans. Sometimes these events are limited to animal to human transfer events only. Other times, the virus has acquired enough changes to be able to be passed from human to human once the initial transfer from the animal occurred. This latter being the case for SARS-CoV-2, the virus that causes COVID-19.